And there is always discussion within the companies themselves, but also within or against or with the regulators. What does these data mean? I mean, up to now, we can't really answer. I mean, it's really a case-by-case thing. Sometimes you simply create the data, and no one really tries to combine them with clinical findings. So I think it's really important, and not just myself, we think that it's really important to investigate the clinical relevance of immunogenicity as far as possible in a general way. We would also like to evaluate, as I said, the predictive value of the existing tools and develop also, if possible, new ex vivo methods. One thing that we were thinking of when we said that was, I mean, there is a lot of T-cell epitope prediction. Some of you might be aware, because this is, I mean, everyone knows that a T helper cell is one prerequisite for a B-cell response, meaning for an anti-drug antibody response. What was not so frequently done was really to look if this really also translates to the T-cell epitopes which are presented in a human setting. So, I mean, really from a patient who makes anti-drug antibodies. So are these T-cell epitopes really the ones that trigger the immune response in humans? And there might be ways to doing that, elude the peptides from the MHC, for example. And so this is the things we're thinking of here. So there might be new tools that we can use in the future for that. We would also like, of course, to investigate in general the immunological mechanism that drive the development of anti-drug antibodies. This is what I said, not just for someone who has a sound background in immunogenicity, but it's also for someone who has a sound background in immunology. Because, you know, for example, I was just thinking, presenting in the thymus, for example, might be something, and the tolerance induction of that might be something that could help us on the long run developing less immunogenic drugs. And of course, one general objective is always to use this data not just for internal purposes or something, but also to be able to tell the regulatory authorities the outcome and to tell them, yeah, there is probably a predictive value behind a predictive tool or behind a combination of predictive tool, and that we probably or hopefully will understand that transient antibodies do have an impact or do not have an impact. So it would also be important for them for the future to evaluate any filing that they will getting. So what is the expected impact on the R&D process? Why are the companies interested in that? Of course, because the investigation of the clinical relevance of the immunogenicity will increase the patient safety and optimize the drug development, because we would be able to answer the question, are transient antibodies really something we should be concerned of? And we could also argue against the authorities on that if we know from a broad basis that they are of relevance or they are not of relevance. So this would really help on first glance. The evolution of the predictive tools could help us in principle if we would find either a predictive tool itself or a certain combination of predictive tools, or even new predictive tools or a combination of all of these things that could give us some predictive value. That would really help us, especially in early drug development, because we could use that as a sort of criteria to select the most appropriate candidates, like PK or like efficacy. So this would be one of the, or could be one of the parameters that drives which candidate will be selected. And if we are sure that this is, let's say, 70% or even more probably predictive, we might, on the long run, end up having less immunogenic drugs, which would, of course, also increase patient safety. And of course, also the investigation of the immunological mechanisms that drive the development might, hopefully, or on the long run, and this is more a vision, lead to something like a sort of biomarker, what we call it here, patient stratification marker, that we can already say why patient X is responding and patient Y is not. And that would, of course, also help us, you know, before we include people in trials, for example, to already know that we probably need to have a closer look at the one and not the other one on immunogenicity. Okay, now coming to the suggested architecture. As we already heard, I mean, this is really just a suggestion. So we sit together and, of course, we made up our mind. But as we just learned, I mean, this is really up to discussion. And if a public consortium would form and say, yeah, we would like to have other ones, or, you know, probably more or less or whatever, it's just up for discussion. What we were thinking of, we have identified 5 packages, and amongst these 5 packages, in principle, package 1 is the first one and also, in principle, the important one for starting of the project. So the package itself is called Identification and Description of Immunogenicity-Relevant Data. What we were thinking of here is we would make some task forces. essentially 3 of them. One is a general immunogenicity task force, so from immunogenicity experts, from both, of course, from the companies, because they might have different views, and from academia and small to medium enterprises. Then a clinical task force, also with participants from both, from the EFPIA and from the public consortium, and a prediction task force, also with participants from both. And the idea is that these people at the beginning of the project should sit together and should first of all define what prediction tools to evaluate, for example, what data, if we think about clinical relevance, what data would be important to be able, from a statistical point of view, to evaluate the clinical relevance. For example, also, I will come to this, which kind of marketed drugs would we use? So how many, which of them, divided into classes or whatever. So these people really drive, in principle, the rest of the call. Because from the outcome of these people, we will then define what to do in the other packages. And the package 2 is intending on the development of the predictive evaluation and development of the predictive tools. Here we're trying to, as we already said, compare with a marketed set of marketed biotherapeutics the different predictive tools. So they will be analyzed into different predictive tools to see if the outcome will be is the same or if there's a lot of difference, and in both in silico and in vitro tools. And not just in these classical ones, but we will also trying to do something innovative, like we just said, for example, trying to elute the peptides really from patients that responded to the drugs and try to sequence them and try to figure out if the T-cell epitopes that have been predicted by the prediction tools are really also presented in vivo. These data, all of these data that we will create, so the safety data, the immunogenicity data, so the results of the binding antibody assays, potentially the neutralizing assays, the result of the predictive assays, the T-cell epitopes, will go then finally in a database that house all of these data, because we need to bring these data together, as we learned at the beginning, because this is essentially missing right now, because we don't have this common view on the data. And then, of course, I mean, the one thing is to put this in a database, but the other thing is there need to be some analysis behind. And that is package 4. So we need to try to use some statistics behind to see some correlation or non-correlation. And then, of course, a package 5, which we're proposing, is also, I think that is in all calls, as I learned, the project management of the project and the communication to the outside and internally. Okay, now coming, I think, to the most important things for you. What are the expected contributions for the applicants? Essentially, our biggest need is really that we have access to patient registries for a selected set of marketed drugs that will be defined by these people. And I just mentioned here some examples, for example, TNF-alpha blockers, interferon beta compounds, and clotting factors. But these are just examples. I mean, we are willing to take everything else which is in the public domain. Essentially because we know that it will be hard to get these data and the samples from the companies themselves for proprietary reasons, for legal reasons, for patent reasons, and so on and so on. And I mean, we still left it open, so it might still be also coming from some pharmaceutical companies, but our experience in the past tells us that this is not really, let's say, feasible. Therefore, we really rely on people that do a lot of public research on these compounds. And if you think about other drugs, for example, the only thing that is important for us is really that there must be a certain amount of immunogenicity data available, because otherwise it doesn't make too much sense. And it would be, in a best case scenario, also possible to still get patient samples, because to, for example, elude the MHC class 2 presented peptides. The other thing that we were thinking is, of course, as I said, I mean, there is a lot of prediction work done in the public domain, in the public consortium, so in small enterprises, small medium enterprises, and also in academia and universities. And of course, our idea is also that you provide these tools, and we will also, with the same set of marketed drugs, use these tools and compare the outcome to the outcome of the tools from the EFPIA companies. one big thing is really the development of innovative methods, so not just relying on the methods that we're already having for prediction of immunogenicity, but also trying to develop new methodologies. And of course, that goes hand in hand with identifying the mechanisms of immunogenicity, because if we don't really know the mechanisms, I mean, then we can't really probably develop new and innovative methods to predict the immunogenicity. The idea currently is also that the database that we need to bring the data together should be handled by the public consortium, not by the private consortium. Yeah, and of course, I mean, in general, we would also like to have intellectual and practical scientific input to address, for example, bottlenecks from the translation of the preclinical work to the clinical data. So what is the things that the EFPIA companies are thinking of in-kind contributions? Of course, we would also provide our prediction tools that we are currently using within the companies, which are quite some. Some companies even use more than one. And we would also apply these tools to the set of marketed compounds that will be selected by the expert panel. As we will do with the prediction tools that are supplied by the public consortium. Of course, there is also some scientific knowledge in the companies for innovative approaches for predicting immunogenicity that sometimes we can't really put into practice because simple lack of resources, because it's not the main focus of a company to develop new prediction tools, for example. But we are, of course, definitely willing to share the things that we already were thinking of, and probably can really then combine together and develop new tools that are more predictive than the ones we're already having. I think what is also very well taken is that the companies have a huge experience in really developing immunogenicity assays in general. So I mean, binding antibody assays, neutralizing assays, and they're quite experienced on how to interpret the data, because they are forced to submit these data to the authorities, and they're quite sure of what is really expected, how the assays have to look like from a quality point of view to be able to compare data to each other. We also have, of course, some experience that we can offer in data management. And of course, there's a lot of clinical experience in general in the companies available that we are also willing to share with the applicant consortium. So what are the key deliverables of the full project that we are thinking of? We hope that we can improve the understanding of the clinical relevance of the anti-drug antibodies, because up to now, there's not really much known on that. Especially, as I said, for antibodies which are just transient or at low titer or not neutralizing. We hope that we can also gain probably some new technologies, not just for the prediction, but also how to detect anti-drug antibodies. So probably new technology platforms that can be used in the future for that. We should establish, or we hope to establish, a database to house all the patient data, the result of the prediction tools, the result of the efficacy outcomes, the result of course, the anti-drug antibody assay results, to bring really that together for the first time and try to gain some statistical evaluation out of that. We hope to get a clearer understanding on the value, on the prediction value of the on the value of the prediction tools, and we hope that we might be capable to really correlate that to the outcome in clinics. So saying that, for example, if we can predict the T-cell epitope with a certain combination of prediction tools in silico or in vitro, that we can also find this in real patients that responded to the drug. Yeah, one thing, I mean, probably you know that in the, for example, in the EMA immunogenicity guideline, it also says a lot about risk-based approaches. So, there are risk it's a higher risk if you administer a protein subcutaneously than intravenously, for example.